Hi, everybody. Good evening. I think we'll start. My name is Elchanan Bornstein. I'm an associate professor at the Department of Genome Sciences and the organizer of this seminar series. And I am very happy to welcome you to our second lecture in the Wednesday of the Genome talk series. Before I will introduce our speaker tonight, I just want to remind everybody that we'll have a Q&A session at the end of the talk. So if you have urgent questions, feel free to ask our speaker. And giving the topic of the lecture today, I'm sure you'll have lots and lots of questions. If you didn't have time to ask those questions at the end, our speakers are always happy to stay behind and chat with the audience. We have refreshment outside to make this even more pleasant, and so definitely keep all those questions and ask our speaker. With that, I'm very happy to introduce our speaker today, Dr. Kelley Harris. Kelley is our most recent and shiny acquisition in the department. She just joined us in the beginning of the year, about 6 or 7 months ago. And I have to admit that when I emailed her and asked her to give this talk, I felt a bit uncomfortable because, you know, the first year as a faculty, you're busy in building your lab and getting students and getting funding and getting the furnitures, and giving a public talk is not always the first thing on a priority list. But then I remembered that the same thing was done to me in my first year. So not only I survived, but that's still something I remember as one of the fun talks that I gave. And I still use those slides to this day, and so I hope she'll forgive me after that. And so Kelley did her undergrad at mathematics at Harvard University, a PhD at UC Berkeley, and a postdoc at Stanford. So he has all the right names on her CV, including an incredibly impressive list of awards for somebody at this stage of their career. Kelley is a mathematical and computational biologist that is trying to develop mathematical models to study genomic data, focusing primarily on understanding the evolution of mutation rates and inferring evolutionary history from whole genome data. And today he'll tell you a lot about your genome and why you're probably a bit more close to this guy over there on this slide than you might want to know. And so with that, please help me welcome Kelley to the stage.
Thanks for the nice introduction, Elchanan. So I am going to talk about Neanderthals and why a lot of us have their DNA in us today. And so basically, most of the stuff I'm going to talk to you about is from this field of ancient DNA that really, I'm going to say, got started just around the turn of the decade in 2010. And it's this pretty crazy field where they let scientists drill into these precious bone specimens and extract the DNA with just slightly more complicated techniques than you might have done to extract DNA from strawberries. So besides this being just really intrinsically cool, it's really fun for me to talk about because this start of exciting results from ancient DNA came right when I started grad school in 2010. So most of the stuff I'm going to talk to you about is, I remember being so excited when it came out and it just being a part of my formative years as a scientist. And it was really great just reliving all that to put together this presentation for you. And the big motivation for studying Neanderthals is they're the closest twig to us on our family tree of ancient hominins. And so today we're up here in the present, going from the past, and we're all by ourselves. But it's sort of an inverse question of looking for extraterrestrial life, of not like, are we alone in the universe now, but were we always alone? And the answer is no, and that we existed at the same time as some of these guys. But even though, given that, we know less just about how we interacted with them and what role they played in our evolution, our history as a species. And so Neanderthals, they actually evolved in Europe, and whereas we evolved in Africa. We both evolved from Homo heidelbergensis in these two places. But then when some humans left Africa and went into Europe, we coexisted with them during the Ice Age. for at least 20,000 years. And so we've known from fossils for a long time just that you would find humans and Neanderthal fossils in roughly the same times and same places. And so it was always sort of a natural question of, oh, what did we do? Did we fight a lot, or did we interbreed, or all of the above? And so there are two hypotheses about human evolution that were debated by anthropologists for a long time, where one of them is called this multi-regional continuity hypothesis, and that used to be the broadly accepted theory. And basically the idea for that is we know that Homo erectus evolved in Africa and then left to go all over the world. But the multi-regional idea is that Homo erectus sort of evolved in place. Asian Homo erectus evolved into modern-day Asians. European Homo erectus evolved into modern-day Europeans, and maybe there was some exchange, but there was this really ancient structure. And that's what used to be broadly accepted in the anthropology community. But then there's this other, what used to be a fringe idea of humans just coming out of Africa relatively recently and just replacing all the Homo erectus that was elsewhere. And then genetic evidence was what let us know that this was a lot more correct. And so the way that people ultimately tested which of these hypotheses was more correct was with some of the earliest comparative DNA studies. And so I'm just going to back up a little bit and talk about two different types of DNA, which is important for just understanding the progression of ideas. So look at a cell. Then the biggest feature in the middle of the cell is the nucleus, and that's where almost all of your DNA is located, about 3 billion base pairs, so like a big 3 billion letter document. But then we also have these mitochondria floating around in the cell that are like your powerhouse or engine that produce the chemical energy that you need. And so these mitochondria, we think, evolved from bacteria that got engulfed by an ancient cell. And they actually have their own genome that's only about 17,000 base pairs long. And because this genome is really simple, it's just inherited from your mother, not inherited by a complex combination of your mother and your father. And the fact that it's small and in lots of copies, this has always been sort of the first DNA that we're able to get, either just out of a person originally and eventually out of ancient DNA. And so looking at mitochondrial DNA is how the first stories about the human lineage got written. And so this is just a picture of what it looks like when when even I go and see human variation on my computer doing research. So you can imagine these as mitochondrial sequences or eventually longer sequences, but basically you'll get a sample from each of a bunch of humans, and almost all of the sites will be identical, line up in a row like this, but occasionally you'll get a site where some people are different from other people, and we call these just the polymorphisms in the DNA. And these all come from mutations, which were just random copying mistakes that happened to the DNA some time ago. And so with this idea in mind, in the mid-'80s, Alan Wilson at Berkeley came up with this idea of the molecular clock, where if you align to the DNA sequences from these people in the present, then you can sort of reconstruct a tree of what their ancestors would be. And then if you have some idea of how fast DNA mutates, like mitochondrial DNA mutating once every 100 generations, then you can even count these mutations and get an idea of how long ago people's ancestors lived. So maybe you have 2 mitochondrial sequences that are different in 4 places, and you say, well, like 4 times 100 is their ancestor was 400 generations ago. And so by making a tree of a bunch of human mitochondrial sequences from all over the world and doing this type of calculation, what Alan Wilson and his colleagues got was a tree with a recent root in Africa, about 200,000 years ago. And the press releases about this said this was mitochondrial Eve, and this wasn't literally like the person who was the mother of all humanity 200,000 years ago, but it does show that this recent out of Africa hypothesis was the most likely. And so we're talking about Neanderthals, and so you probably want to know, where do Neanderthals fit into this? But it was quite some time after this was published before we got to know the answer to that. And so it was 18 years later, this guy, Svante Pääbo, he basically made it his mission that he really wanted to learn about where we came from by sequencing DNA from Neanderthals. And so he got this big institute in Leipzig, Germany, built for the purpose of figuring out how to sequence ancient DNA. And then eventually the first Neanderthal DNA that got sequenced well was from these bones found in Vinja Cave in Croatia. And so, as I described before, their team drilled into these bones and extracted a mixture of DNA that was about 95% sequences from bacteria contaminating the bones, but about 5% DNA that looked vaguely human-like. And they had to search for quite a while to find a bone that was this good, that had this much human DNA. And it's just, concentrating that was key, because if you only have 5% human DNA in your sample, then to sequence that, it's 20 times as expensive as if your whole sample was human. And so there's a fun line from Svante's book about his idea, like why this sample was so good. So he says, oddly, the fact that the Vinja Neanderthals were cannibalized, or at least defleshed by other Neanderthals, may be responsible for the fact that we found rather a lot of Neanderthal DNA and relatively little bacterial DNA. So someone nicely cleaned off these bones and kept them from degrading too fast, and now we are the beneficiaries. And so when we got the first Neanderthal mitochondria from them and compared them to humans and to our closest living relatives, the chimp, you got this nice result of, if you look at human-to-human comparisons and ask how different they are, that's sort of distributed like this. And then comparing a human to a Neanderthal, that's always more distant than a human to a human, but always less distant than a human to a chimp. So that fits this molecular clock idea of us having a much more recent common ancestor with the Neanderthal. And so the punchline of the first Neanderthal mitochondrial DNA sequencing, it was slightly boring, just that Neanderthal variation lies outside human variation. So it looked like this, just when they sequenced about 300 modern humans, they formed a nice tree, and then the Vinja Neanderthal was outside of that. And with a common ancestor about 500,000 years ago compared to the 200,000 years ago here. And so, of course, what we wanted to know was whether Neanderthals and humans interbred in Europe, and that did not answer this question. It's like, maybe, maybe not. But luckily, a few years later, this 2010 paper that I say started ancient DNA being interesting, that's when it was proved pretty unequivocally that some interbreeding did occur. And so the scenario that we think happened is that there was this human Neanderthal common ancestor here, and humans and Neanderthals sort of evolved on different continents. And then when some humans ventured out of Africa, there was some interbreeding with the Neanderthals who were there. And we don't know exactly where this happened geographically, but it might have been in the Middle East, sort of the first port of exit of people out of Africa before they diversified into all these other groups. And so now, and then an interesting thing happened with this was going back to this multi-regional hypothesis and recent migration of Africa hypothesis. Like, now we know neither of them is completely correct, and we like to say it was a leaky replacement of modern human DNA, mostly replacing everything archaic outside, except for a little bit of these leaks. And so I've heard one thing that happened with this is some of the anthropologists who had liked the multi-regional theory and then had to say that they were proven wrong, are now like, oh, now you DNA evidence people proved we were right all along, or 2% right anyway. And so just how do we know from the evidence that Neanderthals interbred with non-Africans? It's, again, just a variant of this clock argument, where if we think about a tree of a European individual, an Asian individual, an African, and Neanderthal, if you sort of think about a hypothesis where this tree was really vanilla and there was no gene flow, then if you trace the lengths of these branches to from the present to the past, basically all the modern humans should be equally distant from the Neanderthal. But it turns out that when you compute these distances from the data, that's not what you get. You get that the African is slightly farther from the Neanderthal. And that signal is statistically significant enough. We know you couldn't get that without the interbreeding happening. And so more precisely, if you're not African, then about 2% of your genome is Neanderthal. And so a surprising finding that happened pretty soon after the initial interbreeding finding is that that East Asians actually have significantly more Neanderthal DNA than Europeans do. And that was the first of many sort of problems with geography, because we've never found a Neanderthal in East Asia. We've found them in Europe, and yet this seems to be the case. And so one idea is that maybe there was a basal Eurasian population of people who didn't interbreed with Neanderthals at all, and then admixed into Europe and sort of diluted the Neanderthal signature there. Or maybe there were just Neanderthals we've never discovered who were in Asia. We don't really know yet. You also have this difference within Africa between the Maasai people who sort of live in North Africa and have probably admixed with Europeans a little bit over time, and other Africans who are more sub-Saharan. So you actually see this gradient within the African continent of the ones who have had more European contact being a little bit Neanderthal compared to the ones who haven't had any. So now I'll just stop and do a little Mythbusters interlude. So one thing, a true statement, is that definitely all non-Africans have Neanderthal DNA, and that it's really uniform within the populations. So if you have a person who's all Caucasian and a person who's all East Asian, then the East Asian almost certainly is more Neanderthal than the Caucasian is. But so just one question we can get out of the way right now is, if you have a weird neighbor who you think is more Neanderthal than you, your intuition probably isn't correct if you think that they're just all Neanderthal. Apparently, Svante gets lots of letters from people claiming their spouse is a Neanderthal. A lot of people get really excited if 23andMe tells them they're in a really high percentile of Neanderthal DNA content. And it doesn't seem, I don't really think it's as exciting as people say it is, because maybe, you have 2.001% instead of 2%. And it doesn't mean that you'll have more Neanderthal brow ridges. And so I think one of the coolest fossils that's been found and has had its DNA sequenced was actually an individual in Romania who had a Neanderthal ancestor just 4 generations ago as like a great-great-grandparent. And so these fossils in Oase, Romania were actually, when they were discovered in 2004, it was fun for me to find this old article describing it, because at the time, I guess people were very much like, ooh, this could be a hybrid. But as I'm a skeptical scientist, if I saw that just based on the fact that it had big teeth or whatever, I'd be like, oh, that's not a hybrid. People are getting excited about nothing. But then, when this 35,000-year-old fossil did finally get its genome sequenced, it turned out it was really a hybrid. And so that's pretty unequivocal evidence of this interbreeding at a time when there were still a few holdouts wondering if it wasn't true. And so how do we know that this individual had a really recent Neanderthal ancestor, and how do we know how long ago our Neanderthal ancestors lived? That actually, we can infer that by looking at how Neanderthal DNA is distributed in our genomes. So basically, if you have this scenario of two human groups starting to diverge, and then Neanderthal gene flow occurring in one, then basically you start off with whole chromosomes coming in from the Neanderthal. But every generation, there's recombination between mothers and fathers, and these Neanderthal DNA chunks get smaller and smaller. And so right now, in human genomes, our Neanderthal DNA chunks are about 20,000 bases long, and that tells us that our Neanderthal ancestors lived about 50,000 years ago. But then this Oase individual had much longer chunks that show their ancestor was very recent. And so there's been a lot of exciting work the past few years on mapping where the Neanderthal ancestry is in modern-day individuals and getting these excavated Neanderthal ancestors. And basically the signal we just look for is if we want to know where the Neanderthal DNA is in some test individual, then we look for places where they look closer to an ancient Neanderthal genome than to an African individual. And basically test that these chunks of DNA are indeed very close to a Neanderthal and much farther than an African. And then training a machine learning model on these criteria, it gets you patterns like this, where you have a real European individual and a real East Asian individual and a bit of their chromosome number 9, and you see all these spikes of regions that are probably of Neanderthal ancestry. And so one thing that we could tell once we started getting these ancestry maps was that Neanderthal DNA has probably been bad for us. There's been a pattern of natural selection to get rid of it from our genomes. And so I know this plot doesn't look like much, but it's basically just plotting the fraction of Neanderthal ancestry in our genomes as a function of how close the DNA is to really important genes. And there's less Neanderthal DNA near the important genes than in regions that we think of as junk DNA. And these dotted lines here are plots taken from our X chromosome. And so we can see the X chromosome just overall has a lot less Neanderthal DNA. And so also genes that are expressed in the testes are super depleted of Neanderthal ancestry. And putting all these things together, some people have suggested maybe that meant that hybridization with Neanderthals caused male fertility problems. And that's a thing we see all over nature a lot, is that when you have a heterogametic sex, like a sex that has an X and a Y chromosome, then they just get vulnerable to fertility problems whenever you have interbreeding between organisms that are starting to become separate species. And so another fun fact that we've learned from a Neanderthal genome is that they weren't very genetically healthy. They lived in these tiny isolated populations all over Europe where they had a lot of inbreeding going on. And so this is a picture of chromosome 21 from a Siberian Neanderthal that was sequenced a couple years ago. And there's this big chunk in the middle with super low genetic diversity. And basically what we know from these chunks of low genetic diversity is probably they were the product of a consanguineous union where the possibilities are that this girl's parents could have been double first cousins or half siblings or a grandfather and granddaughter or uncle and niece. And these all have sort of the same probability based on the lengths of these chunks without diversity. But yeah, shows they probably didn't have a lot of options who to mate with in their Neanderthal communities. Then they perhaps had to turn to us. And this is a map just showing the sites where Neanderthal DNA has been obtained so far. So there's this El Cidrón site in Spain, this Vivindia site that was the first one, this Mezmeskaya site in Russia, and then this Denisova site in Siberia that I'll talk about a little more later. And they're all of different ages and slightly different, distinctive subsets of Neanderthals. And so the Neanderthals found in Croatia actually seem to be the closest relatives of whatever ones contributed to the human gene pool. And so it's possible that interbreeding occurred closer to Croatia than any of these places. So there have been some studies asking, what are the Neanderthals doing in our genome now if they were so bad that we've had this natural selection happening? And so one study, so they actually went and used people's electronic health record data for good science, not for evil. But basically, the electronic health records have codes showing what illnesses people were being treated for. And then when there's genetic information available about these patients, it was possible to look for statistical associations between various bits of Neanderthal DNA and the electronic health record codes. And this is the list of diseases that came out as having, if you have more Neanderthal DNA in specific regions, you'll be at increased risk of them. And so it's kind of funny that tobacco use is on here when probably Neanderthals didn't use tobacco, but it probably just goes in with this suite of neurological disorders, somehow often being the byproduct of Neanderthal DNA. And so this is the only figure I'm going to show from some work of mine, but so I've done some theoretical work, basically predicting that what Neanderthal fitness would have been given their levels of inbreeding. And so under the model that my collaborator and I were using, we basically predicted Neanderthals would have been at about 40% less fit than humans. And so basically when they would start interbreeding, then selection would act really strongly to reduce Neanderthal admixture proportion over time. So we actually would have had to start with maybe 10% of our ancestors in the past being Neanderthal to get down to a 3% level in the present. And so my advisor at the time made me add this dumb sentence to the end of this Q&A on it that one could perhaps argue Neanderthals didn't disappear due to warfare or competition, but due to love. So I mentioned I would come back to this Neanderthal from Siberia. And the thing that's really cool for that is there's just this cave in the Altai Mountains that is somehow really, really good at preserving ancient DNA. And it's probably because this cave is really cold and dry, which is just good for preserving biological specimens. But yeah, there's some magic other formula, because not every place that's cold and dry has produced such good ancient DNA. And I recently learned another fun fact. So the only thing I'd ever learned about the Altai Mountains being good for was this cave. But then this lady who's been in the news lately for being indicted for a Russian spy, I saw one article that she's from the Altai Mountains. So maybe she's actually a Neanderthal spy. So basically, yeah, the bones found in Denisova Cave, there's been this little toe bone and this tiny fragment of a pinky bone. And they actually turned out to be 70% hominid DNA and just 30% bacterial contamination, which is way better than the Vindia bones are. And so, of course, these bones were sequenced in a hurry, and the toe bone yielded a really high-quality Altai Neanderthal genome. That's the inbred one that I showed you earlier. And this is basically almost as good of quality as a human genome that you could get from you or me, which is pretty extraordinary. But yeah, the story was even cooler with this pinky bone, because when they sequenced that, they found something that wasn't quite a Neanderthal. It was a species that had never been described based on morphology before. And so it was named the Denisovans after the cave that it came from. And yeah, it's this dramatic first title, Complete Mitochondrial Genome of an Unknown Hominin. And so, yeah, these guys are really mysterious because we basically only know them from their genome. Just trying to know stuff about them, the mitochondrial and nuclear DNA actually told really different stories here, where basically when the Denisovan mitochondria were sequenced and the tree was made, Denisovans were like way outside of the variation of Neanderthals and modern humans, and we thought maybe this was Homo erectus or something. But then when the nuclear DNA was sequenced, it told a different story of Denisovans being a sister group to Neanderthals, and then humans being over here. And so we think the nuclear story is more reliable, so Denisovans were more close to Neanderthals, but that their mitochondria are so strange, and they have a few, enough strange pieces of their nuclear genome that maybe they also had some interbreeding with some more distant, super archaic hominin. And what do the Denisovans look like? We really don't know much about that, because all we know is their little pinky, which the archaeologists couldn't even tell wasn't a Neanderthal. And since then, we have some of their teeth, and they had really huge teeth. And so maybe it was awkward when there was interbreeding and the children had mismatched teeth for their heads. But yeah, I guess we have to stay tuned for the random sequencing of other bones to see which could be Denisovan and tell us more about what they were actually like.
But do they belong to the same person?
Mm-hmm.
Bone
No, so there are a few teeth that have been found that are all from different people, and then this finger was from a different person too. And so this is another technical plot, but what it basically shows is we're able to take a genome and estimate what the population size of the population of that individual came from over time. And so in the ancient past, all humans and Denisovans and Neanderthals came from the same population, but these plots are the Altai Neanderthal and the Altai Denisovan, and about 500,000 years ago, they started having this precipitous population crash, and it was just a lot worse than this out of Africa bottleneck that happened to humans who left Africa. So I said that there would be more geographical puzzles in here, and one of them is that you might want to know who Denisovans interbred with. It turns out not Siberians. It was Aboriginal Australians and people in Papua New Guinea, even though we have no idea if the Denisovans lived anywhere near those guys live. And this is just a map, showing Denisovan ancestry is concentrated here. And it's also a bit in South Asia, but very little in Europe. And interestingly, people from Papua New Guinea have the most archaic DNA of any of the modern people we've looked at. So Melanesians are people from Papua New Guinea, and the amount of archaic sequence they have per individual is about twice as much as what an East Asian has per individual, which is about twice again what South Asians and Europeans have. And then if you look at people from Papua New Guinea, their archaic DNA is about half Neanderthal, half Denisovan. And it actually appears to have been a different Neanderthal population that contributed to these guys than contributed to these guys. So another Denisovan puzzle, I mentioned we don't know what they looked like, but one random thing we do know about them is they were probably well adapted to high altitude. So I'll back up a little bit about the story of how we know this. So if we look at modern humans, anyone can sort of acclimatize to doing better at high altitude, but there are only 3 populations of people who are genetically adapted to do well, and those are people in the Tibetan Plateau, the Ethiopian highlands, and the Andes. And so Tibetans in particular, their adaptation to high altitude is they don't overproduce red blood cells the way that most of us would do if we went to high altitude. We have to do that to get by, but they somehow don't have to do that. And that means that if they live at high altitude for a long time, so actually Han Chinese people who go live in Tibet are at risk of heart disease because their blood gets really thick, but Tibetans don't have that problem. And also, probably like the key thing that helps their population stay healthy in their environment is they are better at not having miscarriages and low birth weight. So I've learned when American diplomats are stationed in places like Tibet and in the Andes, they're sort of recommended if they get pregnant, they should go home because it's so high risk to have babies in that environment. And of course, if you get better at having babies in that environment, it impacts your fitness a lot. And so the lab where I did my PhD at Berkeley got interested sort of independently of this ancient DNA stuff, of figuring out what's the genetic basis of the Tibetan adaptation to high altitude. And the way they did that was they compared Tibetans and their close relatives, the Han Chinese, and the more distantly related Danish Europeans. And most of the genome, Tibetans and Han Chinese are extremely close, and Danes are a bit farther away. But a few regions look more like this, where Tibetans are the outliers. And so these are maybe regions where genetic change has happened really fast because adaptation is going on. And so there are 3 genes that were outliers on this test, and the biggest one is this gene called EPOS1. And basically, my old labs was studying for a while, like they indeed found that variants in EPOS1 changed how your blood responds to high altitude. It's responsible for Tibetans not overproducing the red blood cells. But this region also looked really weird, basically. Like, it didn't look like what we would expect from a gene that just got a new mutation that was good and that got selected on. And so my friend Emilia, who was studying this, one day, like, she was being frustrated and said, oh, maybe it's the Yeti, and compared the Tibetan EPOS1 to the Denisovan one, and it turned out to match. So this barcode-y thing, it basically, the black bars are positions of genetic variants, and you can see just they don't line up at all between Han Chinese and Tibetans, but then they line up really well between Tibetans and Denisovans. So we think this was a case of adaptive introgression where the Tibetans just got this variant from Denisovans that was useful, and so now like the archaic gene is basically fixed in their population and absent other places. And there are a few other cases of adaptive introgression, genes that from archaic species that were really useful and got selected in various populations. So there's another one in the Greenlandic Inuit that looks like it's related to body fat distribution and just how the fat tissue differentiates and helps them survive in the cold better. And just if you look at Neanderthals, there's weaker but detectable signals of selection in genes related to the immune system and hair and skin and pigmentation. And we think that that might be because Neanderthals were sort of pre-adapted to the European environment for a long time before humans got there, and that since pigmentation and skin genes have something to do with being adapted to that environment, it was sort of easier to pick those things up from Neanderthals than to for humans to acquire them on their own. And with the immune genes, it could be a couple different things. Maybe Neanderthals could have been pre-adapted to some viruses present in Europe, or else just more diversity in the immune system is always good too. And so another thing you can get from looking at these landscapes of Neanderthal and Denisovan DNA is some regions of the genome have been just completely purged of it, like archaic DNA in some regions is really bad. So this is an introgression desert where you see on chromosome 7 like these sort of spikes of archaic DNA present, and these are the Melanesians who have Denisovan and Neanderthal, and then East Asians, South Asians, and Europeans who have just Neanderthal. But it all seems to avoid a few regions like this, and so maybe this was the site of some kind of speciation gene. And there's also this correspondence. This is just like the plot I showed you earlier of the X chromosome and important genes being depleted of Neanderthal DNA. And you see the same pattern with Denisovans, how they were, that DNA seems to have been bad in a similar way. And so one recent finding is that it looks like there was actually gene flow the opposite way too, that this figure just shows, oh, there was archaic gene flow out from Neanderthals and Denisovans to these guys, but there was also a gene flow in where basically we think it was an earlier out of Africa event that didn't leave any descendants who are alive today, but that these were humans who went and interbred with the Altai Neanderthal. And so then if you go and look at that genome, you see little bits of that look more human. And so it wasn't just a one-way street, this exchange. And so you might be asking, was there any archaic gene flow in Africa? Because I just keep saying that they don't have any of these recognizable bits of Neanderthal and Denisovan DNA. And so one unfortunate thing is just like Africa is way more interesting hominid fossils than anywhere in the world, but it's also really hot and sometimes humid, and like those are the conditions that really degrade the ancient DNA that's present in fossils. And so a lot of people are working on getting better at salvaging very degraded DNA because it would have a huge payoff here, but that technology is not there yet. So we can use mathematical models to just sort of just look at the African genomes and ask, is there anything that looks like very diverged and weird? And so some of these studies have said, yeah, maybe there were sort of unknown archaics in Africa that contributed to modern African populations, but we don't know it with the same certainty we know when we can compare it to a fossil. And so I sort of said, oh, it's just like that we don't have the right fossils, but the reality is there's also this persistent racial bias in genetics that has not been getting better fast enough, where even today, about 80% of all the DNA that's ever been sequenced is people with European ancestry. That's even more true of ancient DNA samples, that it's focused on fossils from Europe. And most of the remainder is Asian, and there are tiny slivers of DNA from other parts of the world that comprise most of human diversity. And so, yeah, it's really only the past year or two that I've started to see ancient DNA studies from outside of Europe, and that, yeah, there's a lot more exciting stuff to come now that that ball is finally rolling. And so, like, obviously Neanderthals are really cool and sort of have made this field of gene flow between distant organisms, like, draw more interest than it used to. And that's actually been changing other fields of biology, and just making all biologists think more about this idea of maybe speciation isn't a very neat process, but genetic exchange is, like, really prevalent. And so people who study heliconius butterflies have been finding all these interesting cases of butterfly lineages donating their color pattern genes to other butterflies and creating these mimics that aren't actually that genetically close to each other. And a similar paper about color patterns in pigeons was published just this week. And also looking at things like plants adapting to different habitats. Basically, any time where you have an organism that's adapting to a lot of different habitats, it's really valuable if there can be gene flow to sort of jumpstart the adaptation based on someone who's been in that environment already. And so I'm just going to ask you guys if you have questions now, but if my questions can't satisfy you, there are these two great books that came out pretty recently that tell a lot more detailed story about ancient DNA up to now. So this is Svante Pääbo's own book of his effort to start sequencing Neanderthal DNA. And it's a fun read, but I also have to put a parental advisory for explicit content on it, because he gets a little bit too excited by talking about Neanderthal sex, and he feels like he has to share, like, moments of his own life that he sympathizes with them. And then this newer book by David Reich focuses a bit more on ancient modern DNA, things like the Romanian fossil from 30,000 years ago that have Neanderthal ancestors and are very different from us, but we're still modern humans. And he's able to keep things a bit more PG. So, yeah, with that, I'll just take questions.